Today we're going to talk about enzymes, and the goal of this video is to describe the function of enzymes. So what is an enzyme? It is a biological catalyst. A catalyst is something that speeds up a chemical reaction. An enzyme is also a protein. So putting all that together, an enzyme is a protein that speeds up a chemical reaction. So they speed up chemical reactions specifically inside of cells. And an example would be a digestive enzyme. Digestive enzymes speed up reactions that break down carbohydrates, lipids, and proteins into smaller molecules that individual cells can use. Now, there are different types of chemical reactions that you can speed up. One of them is a catabolic reaction. This is a reaction that breaks down large molecules into smaller, simpler parts. And that's what you see in this image. You have a large molecule. An enzyme is going to break that into smaller pieces. And in the process, energy is released. So energy is an output of a catabolic reaction. The other type of reaction is an anabolic reaction. And this is a reaction that builds large molecules from smaller, simpler ones. A good way to remember this is think of anabolic steroids. You might have heard of that term before. With steroids, somebody's building muscles, right? So anabolic reactions build large molecules from smaller, simpler ones, and energy is required for this process. So in this case, energy is an input. You have to use energy to build this larger structure. Now, how do enzymes work? I'm going to go through three basic steps. The first is enzymes bind to reactants, and these reactants are called substrates. The specific part of the enzyme that binds to the substrate is called the active site, and it's labeled in the image at the bottom of the screen. Now, the enzyme can slightly change its shape to more tightly fit the substrate, and this is called induced fit. The second step is the enzyme has formed an enzyme substrate complex. So they're all kind of one unit now. They're all bound together. The third and final step is chemical bonds are either broken or formed in or between the substrates at that point. So looking at the reaction on your screen, you took these two smaller pieces, they were bound to the enzyme, and the enzyme caused a bond to form between those two. And so now you have this new product. It's a larger molecule than what you started with. So this would be an example of an anabolic reaction. You would have had to use some energy to form this larger molecule out of these smaller units. Now, an enzyme, though, this is important, an enzyme does not get used up during this reaction. And since it wasn't used up during the reaction, it can keep being reused in other reactions over and over and over again. Okay, now, I told you that an enzyme speeds up a reaction, and I showed you what it looks like when it's going through the reaction. But how exactly does it speed up a reaction? Well, specifically, enzymes lower the activation energy. And activation energy is the minimum energy that's needed to undergo a chemical reaction. So in this graph on the right, you can see a blue line, and that blue line is showing you the amount of energy that's needed for this reaction to go from the substrates to the products at the end. And you can see there's kind of a hump, and the hump is how much energy is required to do this reaction, to get it started. But notice the red line shows that the hump is smaller, so it doesn't require as much energy. And that's what I mean when I say enzymes lower the activation energy. Not as much energy is required to undergo the reaction. And why is that the case? Well, by binding substrates at the active site, the substrates are positioned correctly so they don't have to overcome intermolecular forces that would otherwise push them apart. And this allows molecules to interact with less energy. It doesn't take them as much energy to interact because they're positioned the right way. Okay, what affects enzyme activity? So there's going to be three main factors: extreme temperature, pH, and ionic concentration. And each one of these can cause denaturation of the enzyme. And denaturation is a change in the structure of the enzyme that results in inactivity. Basically, once it's denatured, it can't form a tight bond to the substrate at the active site anymore, so it becomes inactive. So to sum up, you've seen what an enzyme does and how it does it.